Hello, everyone. I join Dr. Joseph Gleeson and members of his research team at UC San Diego as they share groundbreaking discoveries from his lab that are reshaping our understanding of spina bifida. Joe's team has identified a common chromosome that significantly increases the risk of developing spina bifida. Even more striking, they found that spontaneous DNA mutations not inherited from either parent can also contribute to the condition. These findings mark a major leap forward in pinpointing genetic risk factors and open the door to earlier diagnosis, personalized family counseling, and potentially future prevention. Dr. Gleeson will discuss how his lab is working to extend these insights across broader patient populations to build a more complete picture of spina bifida's origins. While folic acid remains a cornerstone of prevention, researchers are still unraveling how it works and exploring what additional strategies might further reduce risk. This session will explore how cutting-edge genetic research could transform care and offer new hope for families affected by spina bifida. Now for introductions. Dr. Joseph Gleeson is internationally recognized for his groundbreaking work in pediatric neurology and neurogenetics. He holds the title of Rady Professor of Neurosciences and Pediatrics at UC San Diego and serves as Director of Neuroscience at the Rady Children's Institute of Genomic Medicine, where he leads transformative research into the genetic causes of childhood brain disorders. With a career devoted to unraveling the mysteries of conditions like spina bifida, Dr. Gleeson's lab has identified more than 100 genetic causes of pediatric neurological disease, many of which were previously unknown. His collaborative cross-disciplinary studies have opened new doors to diagnosis, treatment, and hope for families worldwide. Dr. Gleeson is deeply compassionate and committed to translating complex genetic discoveries into meaningful care. Dr. Gleeson is joined tonight by SBA's Medical Director, Dr. Timothy Bry. Dr. Bry, a developmental pediatrician, is recently retired from developmental pediatrics at Seattle Children's Hospital. Also joining tonight is Naomi Meave Okada, who is a clinical research coordinator for studies in pediatric neurogenetics at the Gleeson Lab at UC San Diego and Rady Rehabilitation. She manages the Gleeson Lab's International Spina Bifida Sequencing Consortium, leading patient recruitment, sample collection, and data coordination across diverse populations. With a background in behavioral neuroscience research, clinical psychology, and bilingual outreach, she ensures rigorous family-centered research aimed at uncovering the genetic causes of childhood brain disorders. Welcome to Drs. Gleeson and Bry and Ms. Meave.
Thank you so much, Judy. My name is Joseph Gleeson. Thank you. And thank you, Dr. Bry, for participating today. And thank you, Naomi, for helping with this Q&A session. And we're so indebted to SBA for all they've done as we try to make advances into the causes of spina bifida. And our goal is to develop better treatments, better ways to prevent this condition. We have a couple of introductory slides and to give you an overview of our recent discoveries, and then we're going to open it up for your questions. And Dr. Bry is going to help me with those questions because I'm comfortable with some aspects of spina bifida, but not all aspects, and there are a lot of very diverse questions. Thank you so much for all of your excellent questions. So I'm going to share my screen, and hopefully we'll be able to see this. Are you able to see this now? That look okay? Great. Okay, so this is me. This is my email address. And at the end, there's going to be a QR code if you're interested in participating in the study. So, and we'll make that available to you if you're interested. One of the things we'd like to make sure we get across is that our advances into what's known about spina bifida are very much dependent upon your participation. We're only limited in our capacity to make discoveries by the number of families that enroll in this research. And all we're doing is very collaborative with the entire research community in spina bifida. So we think it's a great opportunity for families to get involved and to learn more about what's going on in the research field. So again, just very high level overview. Most of you are very familiar with spina bifida um, and the associated paralysis of the lower limbs and the association with hydrocephalus. And we know that folic acid has a huge impact on preventing the condition, but it's not a cure and it doesn't completely prevent the condition. We don't actually know how it works. And we don't really understand much about the genetic causes of spina bifida at all. We know there's many different forms of spina bifida. There's the myelose, in which the spinal cord is involved, and there's the meningoceles, where the spinal cord itself is okay. And then there's the, more subtle forms like the spina bifida occultas and the tethered spinal cords. Now, the neural tube, has to close very early during the pregnancy. This happens even before most women know they're pregnant. And this is why folic acid has to be in the diet. Because starting to take a prenatal vitamin after conception is really after all the actions happened. And the neural tube folds like in this schematic and makes this tube. And the heritability of these neural tube defects are about 70 to 75%. That means the genes play a huge role, but we don't know which genes they are and we don't know how they're working. And that's why we have been studying this. We know that there's about 100-fold recurrence risk in families. So after the first child in the family is diagnosed with this, there's a much higher risk of a second child, or that same thing goes for parent to child. But the genetic causes themselves can help us distinguish between which are the really high risk and the low risk families. There's roughly 200,000 patients in the US with spina bifida, and there's over 100 different mouse models for spina bifida. But there's a lot of disconnects because even though these things really point to genes, we didn't know the genes themselves up until our recent publications. Now, there's been a lot of talk about how the genes might influence spina bifida. And up till now, most of the studies that were done were recruiting patients with spina bifida and controls. So most of those were case controls kinds of studies. We learned a little bit, but really, I don't think we can say we really cracked it. So our lab, our research efforts took a very different approach. We decided to study de novo mutations. These are mutations that are not present in either parent, the father and the mother. They're only present in the child. They come up spontaneously. So this was our hypothesis. This was our guess about what causes spina bifida. New mutations present just on one of the two copies of each gene that are not present in the parents. This means we had to change how we recruit patients with spina bifida for these studies. We had to work with trios, which means where we can have sample both the father, mother, and the individual with spina bifida. So we had to start from scratch with our recruitment. And in the result, we have two publications. One came out last year, one came out this year. This paper came out in Nature, and this is looking at the contribution of de novo mutations in spina bifida. What we found was roughly 25, 22% of the patients had mutations that are de novo, and it identified 187 genes. And this was from roughly 850 trios that we were able to recruit. And we credit the Spina Bifida Association with help in recruiting those trios. These 187 genes we can't say they're all causing the condition, but they're definitely risk factors. And we need your help in confirming these results and in making a clinical test for spina bifida, a genetics test. That's one of the things we're aiming for. This was done with the Spina Bifida Sequencing Consortium. Our laboratory led this study, and we had a lot of help from spina bifida experts around the world and clinics around the world. and a lot of researchers that helped us make these discoveries. Second paper was published last year in the journal Science, and what we found, as Judy mentioned, was a very common chromosome deletion on chromosome 22 that seems to increase the risk of spina bifida appreciably. This was completely missed in the past. We don't really know how it was missed, but we can say about 1 or 2% of patients with spina bifida have a de novo deletion of this chromosome 22, and this is highlighted here. That increases the risk of spina bifida. And I'll talk a little bit how we made that discovery and what the implications are of that. These are the two main papers, one in Science and one in Nature. And if you're not in the research field, these two, it's pretty rare that you get a field has a paper in one of these two journals. To have these two papers in these two top journals within like a single 12-month period, really, I think is reframing how physicians and clinicians are thinking about the causes of spina bifida. We've been working a lot with a close colleague, Melanie White at University of Queensland, and she's been modeling some of what we've discovered in quail. These are birds, and it turns out it's very easy to watch the neural tube close. This is what I showed you before in the schematic. These are some of the movies she's able to make about how that closure happens, in real time. And so what we'd like to do is take some of those observations that we've made from patients and now apply them into different models like her model in quail. To make these advances, we worked with international colleagues to develop the Spina Bifida Sequencing Consortium with the support from the Spina Bifida Association. This is a kind of a clearinghouse where we can enroll patients, enroll families, and broadly share between researchers. So you don't have to donate samples to study in this place or that place. You can just do it one time. If you agree to participate in this effort, we agree with the rest of the members of the consortium to share data privately within the consortium with the members. And this is really going to help, I think, advance research in a way we weren't able to do before. Folic acid, we know, makes a big impact in reducing the incidence of spina bifida, but we don't know how it works. And so we've been applying folic acid now to some of the models we're developing. For instance, the quail model, some mouse models we've been making now to study some of the mutations that we've identified. And we're hoping to have a clear idea about how these things are functioning together. The consortium so far consists of maybe 30 or 40 sites around the world. We have several sites in the US and in South America, and lots of different countries in Europe and in Africa and in Asia. And some of these countries have folic acid in the diet and some of them don't. So we're paying close attention to this, how these, what their fortification policies are. We're recruiting trios as much as possible. And then we're doing what's called the genome sequencing. This is where we sequence all the bases in the genome. And we can identify then those de novo mutations that pop up just in the baby or in the individual with the condition and not present in either parent. And we're putting those in then to a very large bioinformatics effort that uses a lot of supercomputers. And now the numbers are crunching and using a lot of AI actually to help us identify these mutations. From the first study that I mentioned, we used the cohort of individuals with spina bifida. Here we call them MM because they have myelo, and a control cohort of roughly the same size, 800 versus 730. We did the genome sequencing and then we compared the results. And I'll just show you the way we think about them is in terms of this pyramid. So the pyramid, the bottom level of the pyramid shows that the cases and the controls have the same number of DNA variants that don't change a protein. But as we go up in severity of the types of DNA mutations up to the tip, which are the most severe mutations, now we can see there's a giant excess of these what are called LGD mutations. These are the likely gene disrupting mutations in cases that are hardly ever seen in controls. So you can see this ratio here is almost a 3-to-1 ratio. This tells us that around 25 to 50% of the damaging mutations that we find in myelo patients contribute to the risk and that with a cohort of around 800 like we had, we should find 10 to 20 recurrently mutated genes. These are genes that we have a high confidence in that cause the condition. And this is roughly what we found. I won't go through the numbers. Where we are right now on this curve is that we have roughly from our cohort of around 800 trios sequenced, we found roughly, as I mentioned, 187 genes, and about 5 of those genes are recurrently mutated. Where we'd like to get is a cohort of roughly 3,000 or even up to 5,000. And these are our expectations. We'd expect to get up to mostly, maybe around 500 genes and maybe 150 or 200 genes that are recurrently mutated. That would give us possibility of really developing a clinical test that could be used throughout the world for pregnancies where we think there could be spina bifida, to make better diagnosis and to make individualized, personalized therapy possible for patients with spina bifida. This is where we're going in the future, and this is why we need your help. Right now we have roughly 1,000 trios. We need to get up to 3,000 for sure. As I mentioned, the chromosome 22 story, we had this chromosome 22 deletion that we found completely by chance. We were not expecting it at all. This is chromosome 22 laid on its side. This is now a zoom in, and what we found is a deletion of chromosome 22 that was present in a lot of the families that we had recruited for this study. Chromosome 22, this region, is the most common chromosomal abnormality in humans that it's clinically relevant. It's present in perhaps 2% of all humans. What we found is there's a huge enrichment in patients with spina bifida with this chromosomal abnormality. But surprisingly, it was not the region that's most commonly implicated in other neurocognitive abnormalities like schizophrenia, which is down on this part of chromosome 22. We found a completely unanticipated common region on chromosome 22 between regions C and D that's not been implicated previously in human disease. This really focused our attention on this region down here. And from this region, then we collaborated with the Children's Hospital of Philadelphia, where they have a large cohort of individuals with this deletion. We asked them if any of those patients has spina bifida or myelo. And they said, no one's asked us that before. But they went back and checked, and they found a huge excess of patients in their cohort with spina bifida. That told us that if we recruit patients with spina bifida, we'll find a lot of chromosome 22 deletions. If we recruit patients with chromosome 22 deletions, we'll find a lot of spina bifida. And that tells us that association is very strong and it's reciprocal, and tells us that this is really an important contribution to the risk. Now, within this region, C to D, there's 10 genes. We didn't know which was the right gene, so we knocked them all out in mouse, and we found one gene in particular called crackle, which is here, shows a range of neural tube defects, and that high folic acid used with these crackle mice can rescue the neural tube defects. So this is the direct connection between human risk for myelo and the role of folic acid in preventing the condition. We need to understand more about this. We're still scratching the surface about the mechanisms, but now we feel we're on the trail of some really important biology. So to summarize the slides here, we found de novo mutations account for about 20 to 30% of the risk in myelo. We discovered 187 genes. About 5 of these genes are seen in more than one family. We need a lot more families to confirm the results, and our goal is to develop better treatments and preventions. These are the folks that contributed to the project, and this is the Spina Bifida Sequencing Consortium. If you're interested, you can use your smartphone to go to this site here, and this will bring you to a website that'll give you a very quick overview of the Spina Bifida Sequencing Consortium. You can enroll here. If you give us your email address, we'll contact you and invite you to participate. You can learn more about what the Spina Bifida Sequencing Consortium is doing, or about our lab website and our funding from various organizations that's allowing us to do this work. So thanks. I'll open it up for questions, and like to turn it over to Dr. Bray to see if how we'd like to take the direction of the conversation now.
Thank you. Juanita will be reading or working with us on some of the questions that came in prior to this community session. And as she covers those, I think Dr. Gleeson will be looking to respond first. And then if I have any additional things to say on those questions, I will. There are certain questions that he might not have the experience or information to answer that I will attempt to then answer as SBA's medical director. So,
Thanks so much. I have the list of questions. If you want, we can go through a couple of them now. And um, one of the first questions was how can participating in this study benefit me or my child? And that's a great question. I think that, this is a research study. It's not a medical test at this point. There is no medical test for spina bifida. What we're asking families to do is help us with this research, help us help the community. But participating can still have a meaningful benefit for families because you'll help advance the science, and the consortium will get back to you with anything that we find and let you know if the results are positive for any of the genes that we found. And importantly, it's unlike a normal test. If you get a test done, you get the results, that's it, the test is over. If you participate in this consortium, we keep looking at the results month after month, year after year, until we find the cause. And we think we're going to find the cause in most or all families. We just don't know what to look for right now. But as we get more experience, we will learn, and then we'll get back to you. We call families up sometimes 2, 3 years after they first enrolled, and we tell them, we found something. And then we're here to answer questions and to give you the relevance of what we find.
I also think that the importance of the studies are research is really looking at, particularly in these early formative stages. I will say formative because we don't have yet a gene pool of genes that we can draw blood or take a swab and say, oh, you have this mutation, so your risk of having a child with spina bifida may be X, and that kind of thing. But it benefits the community because the more we learn from research, those kinds of tests can be developed. And allowing families to know in advance what their risks may be, and get information with regard to that.
Exactly. And some people ask, will the research ultimately lead to the ability to alter the affected genes? It's a very good question, but we hope we know we can't go turn the clock back. That's just reality. What we're hoping is that with these genes, we can develop better anticipatory guidance. Or for instance, one thing we're finding is if you have like certain gene, you might be at higher risk for a certain condition within the myelo spectrum. Like who are the ones that get the hydrocephalus? Who are the ones that get the bladder infections and those kinds of things? Who are the ones that eventually have other organs involved, like heart or eye? But I think what's even more relevant to the community is that we hope to have developed better ways to prevent it. Folic acid really was identified just by chance. Really, no one had any idea. But we think we can do it better in the future. Through finding the genes, we want to find those vulnerable points and develop maybe even a better nutritional supplement or a vitamin or something that can help prevent it even more powerfully than the effect of folic acid. Another question we get a lot is, how might the work impact future treatments of spina bifida? And we think that this kind of work, and it's not just us, it's really the whole research community. There's a whole group of people across the whole world at different universities, academic medical centers, and the pharmaceuticals industry. They're all interested in preventing spina bifida. And we're all trying to understand the causes so that we can impact future treatments. The future treatments could come in many forms. We're seeing a revolution in personalized medicine now. It's really almost unimaginable, the kinds of things that could happen in the future. CRISPR, gene editing, personalized medicine, and things like this. We don't know what's possible, but one thing that's very clear, we need to learn more about the causes of the condition before we can really expect to reap the benefits of the revolution that's happening in biomedicine and preventive personalized medicine. There's a question from one of the participants who said, I was one of the people who participated in the study. I'm glad I did and glad to help the advancements related to spina bifida. Well, I'd like to thank you very much, Milton, for that. That's fantastic. We have, like I say, almost 1,000 families that have participated. And when a family does participate, they contact us. We can't contact you. The Spina Bifida Association can contact you, but it's up to you to contact the consortium to let us know you're interested. And that's just the first step. We give you information then to educate you about the study, the risks and benefits of the study. There's very few risks, and we hope that the risk-benefit ratio is favorable to you. What we do, if you did contact us, we would have a short Zoom call, discuss the study. If you're interested, we'll send you an envelope in the mail, and that would have some little plastic tubes to collect a spit sample from father, mother, and affected individual. And you return those to us in the mail. Everything's done in the mail and over Zoom, and there's no cost. And it's approved by the ethics committee at the University of California, and funded by the NIH. So we are in a position now to really make advances that we weren't in the past. And we're happy that we have positive experiences from people who have participated. We can't get back to everybody with an answer. That's important. I think about 20% of the time we can find what we think is the cause. We get back to the family or to the doctor, depending upon what you request us to do. And in those that don't have an answer at the time, we keep looking, because the technology keeps improving. The AI we're applying now to the genomes, it's solving more and more of the cases. And we think it's, going to continue to unfold like a Sudoku puzzle. If you've done one of those, you get to a certain point when you can kind of see what's happening, and you can pretty quickly, all the answers fall into place. We're not there yet. We're still kind of at the beginning, but we can already see some of the patterns starting to emerge, and it's really exciting. There's another question here from Carrie. Could you use two spina bifida parents with a healthy child? Oh, now that is a very good question. We are mostly recruiting. Let me try to answer that as best I can. We're mostly recruiting trios. Trios is the ideal for us because we can find those de novo mutations that I mentioned. We're starting now, as more of these pieces are falling into place, like the Sudoku puzzle, we're starting to recruit individuals with the condition, because we can already now make sense of the genome where we couldn't before. So if you're an individual with myelo or some form of spina bifida, please get in touch with us and we'll work with you. Maybe some people have just one parent surviving or no parent surviving, or something like this. We'll work with you and see what we can do. We'll probably offer to invite you to participate, and then we set the priority on who we're performing the genomes on, depending upon which patients, which families are the most likely to yield the next breakthrough. But I think it's a great time to get involved. Dr. Bray, would you like to choose one of the questions, or?
Sure. Let's see. Let's see. Chances of having a child with spina bifida when both parents have it, and That's a good question. We know statistically that individuals with spina bifida have an increased risk of having a child with spina bifida. So rather than the 3 out of 10,000 births, that risk probably goes up somewhere between 3 to 5 out of 100 births. I may have that number a little bit off, but it certainly is a significant increase over the general population. Similarly, siblings of individuals with spina bifida, where the siblings don't have spina bifida, also have potentially a 3 to 5% risk, 3 to 5% risk of having a child with spina bifida, which is again higher than the general population. That is one of the things that really, I think much earlier on than and much older than Dr. Gleeson is, led us to really understand that there were genetic contributors or genetic factors which contributed to spina bifida. And I think with the advances over the last 10 to 15 years, have allowed us really to get to the genetic causes. Because the genome has been sequenced, we can now look at individual genes, where 15 or 20 years ago, that technology was not available or was very rudimentary. So there is an increased risk. That's also why understanding these genetic mechanisms is important. So it's again not necessarily important. It may be important for the families of who have children with spina bifida. It's also important because siblings of children with spina bifida who don't clinically have spina bifida themselves may also have an increased risk of having a child with spina bifida with their partner. And so we need to keep looking at the genetic mechanisms and understanding those, and looking at the mechanisms and the inheritance patterns of those mechanisms.
Yeah. If I could just add one, just to put a little finer point on that, Dr. Bray, I completely agree with you. Those numbers, they were developed empirically, which means just from experience and pooling all the different causes together. Now that we are able to start to make out the individual causes within a family, within an individual with spina bifida, we can give them a much more precise risk. So we know, for instance, if you happen to have the chromosome 22 deletion, which we know is the most common genetic form of spina bifida at this point, present in 1 or 2% of all cases that we've recruited, we know you have about a 14-fold risk. That's a much more precise risk estimate. And that can help families make decisions about next pregnancies. They can get their healthy siblings in the family tested, because it's possible they're carriers, like Dr. Bray mentioned. And so I think this is going to start to really unfold more and more as we learn more. Excellent point. Thank you, Dr. Bray.
We do not have, so one of the questions was, would having a male partner take folic acid prior to conception also assist in preventing? We don't have any literature on that information specifically. The folic acid information related to mothers, fathers were not included in those studies. And we don't know whether it is likely the folic acid in combination with genetic effects or potentially other environmental effects. So there may be such things as mothers who need to take anti-seizure medications, or mothers who are significantly overweight, where there may be increased risk factors that may or may not be related to genetics, but to the intrauterine environment or the maternal metabolism in some way, shape, or form. So we don't know. We don't know of any particular health risks of taking folic acid or making sure that your folic acid levels are in the normal range as a male. So but we don't have any evidence that suggests that a male partner taking folic acid changes the risk at this point in time.
Dr. Gleason, I know you mentioned like the benefits of the study, but could you talk about what the benefits are to adults with spina bifida specifically? I feel like the more obvious benefits are for children with spina bifida or like future children with spina bifida.
Yeah,
What are your
I think
thoughts?
it depends on how, and that's a great question. Yeah, I mean, I think what we're mostly uh spina bifida is a lifelong condition, and there's a lot of adults with the condition. We would welcome all. The thing about your genes is they don't change over your life, and we would be just as happy to enroll a trio in which the affected member is a child or an adult. Or if you're an adult with spina bifida, please get a hold of us. We would love to enroll you and talk to you about the study. You can help us prevent the condition in other people and help us develop better treatments. We don't know which form they're going to come. We don't know if there's going to be a better treatment for spina bifida in the future that can benefit adults or kids or where that breakthrough is going to come from. But we're scratching the surface of the causes, and we think that's really important. I think for identifying genes, let's say, that are putting people at risk for other conditions, we have several examples of families that we've done sequencing on, and we found out that they came to us with spina bifida, and we found out through their gene sequencing they actually have uh, um, a genetic cause that links it to another condition that then we can do anticipatory guidance through genetic counseling on them. For instance, they maybe have a higher risk of deafness. They have spina bifida and a higher risk of deafness because they have this gene mutated. Well, that can help the families. That can help the patients. So there's lots of ways that this kind of study can help the enrollees. One of those questions I saw go by on the chat was, are we worried about the NIH and the funding situation? It's a complicated question. I think, just in the last 6 months, we have seen kind of a lot of changes at the NIH and funding for research, and we're working with advocates on the Hill to help ensure funding for important research questions. Everyone agrees, no matter what your political affiliations are, that research into conditions like spina bifida is important. There seems to be quite a lot of unanimous support for this. And we saw just last week that the NIH is very supportive of maintaining the NIH budget. Sorry, Congress is very much in favor of maintaining the NIH budget. So we're hopeful that we can continue to make the case for strong support for research in spina bifida.
For those of you who might not be aware, the president's, so in March, there was a continuing resolution passed so that funding to the NIH, as well as the CDC and the National Spina Bifida Program at the CDC, continued funding at the 2024 levels. That funding runs out at the end of September 2025, and Congress is taking up the president's budget proposal for FYI 2026. And I just looked this up because I had rough numbers in my head, but I wanted to look it up. So the president's budget recommendation, I'm sorry, I'm looking at a variety of papers here.
While you're looking that up, let me just mention one other thing. For every dollar we receive from the NIH, we get a multiplier in the form of a program from the NIH called the Gabriella Miller Kids First. So Congress had a couple $100 million left over from one of their taxes they had. And it was sitting around, they didn't know what to do with it. And there was an opportunity to fund research into structural disorders. Spina bifida is one of them. Congenital heart disease, those kinds of things. And that program is called the Gabriella Miller Kids First program. And we take full advantage of that. That gives us the opportunity, and that's locked in place. That's separate from these ongoing discussions because they just approved another, I think, 5 or 10 years of that. We're very fortunate. So if you enroll in this study separately, the government is funding the generation of the genomes. This gives us a multiplier. So it's an amazing time to enroll. We never had this opportunity before.
So the president's 2026 budget proposed cutting the National Institutes of Health funding by approximately 40%. Now, the Senate recently provided significant support and recommended and passed a resolution, actually, that supported the NIH's funding at slightly above the current level, but certainly not slashing the funding dramatically. That has yet to be taken up by the House of Representatives. So there is still a process to go through, but there is indeed support from Congress. They're hearing from their constituents that funding for medical research is important. Research all across the board. So not just for spina bifida, but everything that the NIH funds, the government funds. So it's also an opportunity for advocacy for your legislators to reach out to them with regard to funding for biomedical research. So I'm sorry to go off on a tangent there.
Well, it's important. I think it's for organizations like Spina Bifida. Definitely. There's a question. Is there a link between spina bifida and genetics of corpus callosum? Obviously, both neural tube defects and known genetics on them. So yeah, there's links for sure. Multiple links actually. And we think this is where the genetics is going to help us because there's about a dozen patients in the cohort that have both spina bifida and corpus callosum abnormalities that go beyond the ones we typically see with the hydrocephalus. and we're looking at those, definitely. We hope to make those links and establish those genetic links, and that'll give us yet another important angle.
There's a few questions that have been repeated about participating in the study. One of them is, do you get the results back if you participate? And yes, you do get the results back. If you have participated, I encourage you to reach out to us at, and you can find our contact information everywhere, but it's, I'll type it in the chat right now. If you want to participate, I just sent the link tree if you missed it in the talk that Dr. Gleeson gave. And then the thing I do want to point out about results is they take about a year. And I don't know if Dr. Gleeson wants to talk more about that.
Yeah, thank you so much, Naomi. As I mentioned, this is a research study we're doing, so it's not a test. And we have to do everything in a massive way. So we collect about 1,000 individual DNA samples and we put them in a giant, machine that generates the data. And we have to put in about 1,000 at a time for the system to work efficiently. So we recruit over the course of about a year, and then we start the next batch of samples. It takes about a year for them to run through the whole process, to do all the quality control, do the interpretation, all the AI. So it takes between a year and 2 years for us to get back the results. That's before we can even tell you if we found anything. So, but we're happy to take any questions anytime. If you're wondering if any of the results are back, if any of you have participated and you want to know, please just contact us. I see Naomi just put in the chat our link there, something called Linktree, LINKTR is the link to the study, and from there you can find our email address. Was that the question that? Oh, there was another question I just saw come up about MTHFR, right? That one came up repeatedly, and Juanita, thank you for flagging that. MTHFR, a little bit of background that people that don't know about it. So we know folic acid is really important in preventing spina bifida. And there's an enzyme in our bodies that converts the folic acid to the active form. And there's very common genetic variants in that gene. And there was great expectation that this was going to be the important cause for spina bifida, this enzyme in this gene and these variants. It turns out that this gene does play some role in risk, but not nearly the role that it was anticipated or that I think some folks attribute. So if you have a variant in this MTHFR, the common ones that are present, I think something like 20% of the general population, it does increase your risk, but only slightly, not to near the degree that we're seeing with these, numbers of genes that we just found. And we think about that in terms of what's called the odds ratio. This is if you have this variant, what's the chance you're going to have spina bifida? If you have that chromosome 22 thing, as I mentioned, you got about a 20-fold increased risk. If you have this variant in MTHFR, you maybe have a 2-fold risk, something like this. It's a modest risk. And there's lots of people who have variants in the MTHFR that are fine. In fact, homozygotes for those MTHFR variants are by and large healthy. So we can't say that this has a cause and effect relationship. But what we're going to do is connect the MTHFR gene to the other results and look at a kind of multifactorial model. That's what we need to build next and hope to have a much more precise risk calculator that's at the individual level that takes into account the MTHFR and all the other genetic risk factors.
I
Sorry.
also think that there's I don't want to say a misconception because I think it was suggested in years past, but currently we know that many people who have the MTHFR variant may process folic acid that's occurring naturally somewhat more slowly, but they can process it. So there have been people who've been recommended to be on folate rather than folic acid. That's currently not a recommendation because we know that people with the variants can indeed process folic acid, but the rate at which they process it may be slightly different. So it's important to discuss this with your providers in terms of whether this is something you need to consider. I will also say that in terms of folic acid supplementation, currently in the worldwide, it's extremely variable. Currently in the US, we add folic acid to wheat products. And I'm off the top of my head, I'm forgetting whether it's passed Congress to now do corn masa as well. But remember that benefit only happens if you eat wheat products. If you make yourself gluten-free, you're not getting it from the diet per se, because it's supplemented in wheat. Also, many products which are organic don't add the folic acid fortification. So that is something to remember as you're looking at diets and what you take for diets. And again, it is recommended by the CDC that all women of childbearing age be on a daily multivitamin, since approximately 50% of births in the US are unplanned, meaning we don't know when they're going to happen. Families may be trying, but we don't know when it's going to occur. So it's recommended that women take folic acid through childbearing years. And that if you have spina bifida or are a first degree relative with spina bifida, that you look at contacting your doctors at looking at doing higher fortification of folic acid prior to conception. And I wanted to make that little plug.
Yeah. Thank you.
Um, I just
question.
wanted to just jump in quickly. Sorry, I know that there were a couple, there's so many questions, but one just popped in about if we've noticed the gene mutation for POTS, MCA's EDS, and is there an increased risk for someone with spina bifida? My daughter with spina bifida has developed POTS and MCAs, and we wondered if individuals with spina bifida are at increased risk for this.
Do you know, Dr. Bray?
I haven't heard of anybody else with spina bifida who has that. So I don't know that there's an increased risk.
One of the questions that I think I maybe saw more than once, or there seems to be some concern about the research results leading to increase in abortions. Well, I just want to make perfectly clear, we are not interested in any of those kinds of aspects. We're not doing research on that, and we don't expect that our results are going to impact that. And anyway, those decisions are outside of our research world. Just to make perfectly clear, we're interested in identifying causes for spina bifida that can help individuals with spina bifida. That's our goal, to help improve care, to help improve treatments. We publish the results in international journals. What doctors do with that information, it's up to them and their interactions with families. But just to let you know, there is no gene for spina bifida. At best, there's going to be risk factors, and it's impossible to use a risk factor to make a determination on an abortion. So we don't think our results are going to directly impact abortions in any way. We hope that this risk calculator I'm mentioning does give individuals with spina bifida a much better sense about their individualized risk pattern, their risk for having future children with spina bifida, or within individual families, or if a parent has spina bifida and wants to have children. That information can help them make their decisions, educated choices.
And at SBA, we believe that decisions with regard to children who are prenatally diagnosed are decisions that should be made by the families and their physicians. Having said that, we also recognize anecdotally that many families get misinformation about spina bifida and what's involved from physicians at times, from what they read on the internet. And so SBA is actively working to make sure that there are resources for for families who are prenatally diagnosed to make a much more educated decision about what their choices may be with regard to that pregnancy.
Yes, and we're of the mindset, we were educated on this very early from the spina bifida community. The spina bifida is not a disease. It's a condition. It's a disability. And there's lots of healthy people with this. We made the mistake one time of calling it a disease, and one of the spina bifida individuals set us straight, And we completely buy into this. This is a condition that we hope to prevent. But and I completely agree with the SBA's approach to this.
There's a question that came up on, is there any research on having a child with spina bifida after having a child without spina bifida? There is not, but we know that there are, again, from Dr. Gleason's work, many potential gene mutations which can occur. And it should be remembered that gene mutations can be de novo or new from some of the gene associations he's making, such as the 22q. There can be genetic variability because it's not the genes from one parent alone, but genes from both parents. So it's very complex. And I will say in my own case, my older brother did not have spina bifida and I do. So every pregnancy is every pregnancy is considered to be the same risk until you have had a child with spina bifida based on what we know scientifically now.
I know we're almost at the end. I just want to thank all the folks that have tuned in and all those individuals that have enrolled in our study. You are making a huge difference. You're really helping researchers around the world build this consortium and make it research advances. It's helping us crack the code. I think we're going to see another wave of discoveries in the next couple of years. And we'll do our best to answer all the questions if we can. If you'd please just email us. If you have any questions we didn't get to, please send us an email and we'll respond individually to every single one of them.
Or
And
you
we
can get them.
send you the questions. Yes.
You can
or
send us the
send
questions.
them
The problem is
to
that we can't respond directly to those people. If you want to,
Okay.
So send them to Juanita, who can then forward them to us and we can look at responding. I do want to make one quick comment because a number of questions came up with regard to this about stem cell therapies. Currently, so I will say that the information is not there. There are some stem cell therapy is still relatively, despite what you hear about it, still relatively in its infancy. Much of the work that has been focused around stem cell therapies have been focused on diseases where there is one specific genetic mechanism. And those diseases were fatal, meaning children died from them, and they're trying to do stem cell or other novel therapies with regard to those. There is not any research that I could find that's on the clinicaltrials.gov website that relates to stem cell therapies in people who already have spina bifida. Some work has gone on in similar disorders such as or other neurologic neuroscientific disorders such as cerebral palsy or spinal cord injury, but those are very much in their infancy and not yet available for clinical use. They're still in clinical trials. Pretty much all of those that I was reading. So it certainly is a treatment for the future, we believe, and we think that it can make some difference, but there is no therapy right now. Just as I will say that prenatal surgery, fetal surgery for spina bifida definitely can help with some aspects of what people with spina bifida go through and potentially improve some aspects such as decreasing the risk of hydrocephalus. However, fetal surgery is still not a cure for spina bifida. And I think looking at treatment interventions from stem cell therapy related to various aspects of spina bifida, such as mobility, are many years off yet.
Dr. Gleeson or Naomi or Juanita, any last comments?
I just want to thank everyone for listening. I'm on the email right now and I see incoming emails, which makes me very happy. I'm so glad you guys are reaching out and interested, whether you participated in the past or are interested in participating. So yeah, thank you very much.
yeah, thank you. Thank you all for participating.
And we've appreciated this, and to our audience, thank you for joining. And you'll be hearing more and more about this research. And if you have any questions that we can help with or connect you with Dr. Gleeson or Naomi or Dr. Bright, please let us know.